My name is Erzsebet Vincent, and I've been working with Viva Horowitz and Professor David Oshalom on the optical properties of cephalopod skin. So what are cephalopods? They're actually the family of organisms that comprises octopuses, cuttlefish, and squid. And while most of us know them better for being sushi or predictors of the outcome of the World Cup, cephalopods actually have a much more interesting and more unique trait. They are masters of disguise. I call them chameleons of the sea, but actually chameleons should probably be referred to as cephalopods of the land because cephalopods are so much better at it. I'll give you an example. Take a look at this coral right here. Now take a closer look. Now take a closer look. This takes place over a matter of seconds. And indeed, octopuses and other cephalopods are able to do this because they have numerous techniques that enable them to change their appearance. One of these, and the one that I'm focusing on, are chromatophores, which are sacs of pigment in their skin which are able to expand and contract over an order of magnitude in size in order to help the animal conceal its appearance. So when we look at these systems, we can look at them from a biological light, but we can also ask ourselves, what could harnessing this type of system do? What kind of applications could this have for engineering, physics, optics, so forth? Now, one of the major possibilities that could arise from this is the idea of adaptive optical technology. And probably one of the perhaps most widespread interest-worthy ideas is that of dynamic camouflage, which is essentially the idea of being able to disguise something much like the cephalopod here disguises itself. Other possibilities are amazing innovations in optical technology such as filters, which might actually be able to change which wavelength of light they are filtering using technology based on this type of system. So essentially my project is an inquiry into what is essentially a unique sort of material. What I'm doing is looking at cephalopod skin as a material and trying to characterize some of its properties with relation to light. So what I've done so far is take a look at the optical absorption, which essentially is talking about what wavelengths or colors of light that the skin absorbs as opposed to transmitting or reflecting. Additionally, I've been looking at fluorescence at a variety of excitation wavelengths. Now what this means is, what the concept of fluorescence is that one wavelength or color of light is used to excite a specimen, which then produces another different wavelength or color of light in response and emits that. Additionally, I've been using photoluminescence to look more particularly at the differences in the skin, the difference between chromatophores and other parts of the skin, and therefore looking at the different structures. So one of the initial things that I did first was the preparation of slides. The first part of this involved removing the skin from the squid, and then preparing it by pinning it down and bathing it in artificial seawater to keep it fresh. I then would excise pieces of skin that contained chromatophores and mount them on slides, creating a wet mount slide using either a few drops of artificial seawater or possibly artificial seawater containing potassium chloride. Now, why use potassium chloride, you ask? Well, it can help you turn this into this. Now, this is actually the exact same piece of skin. And what happens is that the potassium chloride causes the muscles in the skin, which connect to the chromatophores, to contract, therefore pulling them open and making them much more apparent. So, what we first look at now are the absorption spectra that I was able to take from these samples. So what we can see here is essentially there are two different samples of squid, which were different specimens of squid that I made slides from. And I've color-coded them accordingly so that you can see that overall, the squid patterns are pretty consistent. You can see that they have a higher absorption here down in this, in the purple, blue, and green visible range, which implies that they absorb those colors and that they would therefore appear more reddish or brownish in color. Now we can actually contrast that with the blanks that I created, which are essentially controls containing seawater as well as seawater with potassium chloride. And you can see that overall they're mostly flat, and if anything they show more absorption in the reddish side of the spectrum, which implies that they would appear more bluish. So it's clearly apparent that the absorption spectra that we see from the squid is very distinguishable from the absorption spectra presented by the controls. Next, I took some looks at some of the fluorescence emission spectra that were created by using different wavelengths. This one is the the emission spectra created by exciting at 250 nanometer wavelength, which is in the ultraviolet range. And what you can see is that pretty consistently the squids produce a curve that looks like that, which does not really look anything like the blank curve, which looks like this. This implies that this curve is actually unique to the material and is not caused by a different factor that would be in the solution that contains it, nor is it also additionally caused by a consistent artifact. So because it appears so consistently, it looks like this is a feature of the skin. An additional excitation wavelength which I used was 300 nanometers, also in the ultraviolet range. And again, we see that the squid have a consistent pattern that does differ drastically from the pattern that is presented by the blanks. So we see again that there is something interesting going on here, that these pieces of skin are indeed fluorescing, and they're doing so in a different way than anything that contains them, which implies that it is a property inherent in the skin. Next, what we did was we used a laser to take some confocal photoluminescence data. And actually what this essentially does is create an image based on the photons that are caused by being excited by a particular laser. So what this image shows us is that when you're using a 532 nanometer excitation laser, we can see that this chromatophore, which is right here, is much brighter than the surrounding skin, implying that it's fluorescing a lot more. Note that the color doesn't necessarily mean that it's fluorescing blue, it simply means that the number of photons is much greater than the surrounding skin. Now you'll notice that it seems that there are some artifacts or lines through the chromatophore right there. These are actually caused because when we were testing the squid skin, it was so fresh that the sample was still moving in response to the excitation by the laser. So, so you can tell this is pretty fresh skin. So this is clearly a property that is present in very fresh skin and is not caused by the decomposition of proteins within the chromatophore. Now we repeated these experiments using a 500 nanometer pulsed laser, and we got similar results showing that the chromatophores were again fluorescing a lot more than the surrounding skin, implying that this is something that is consistent with both of these wavelengths of excitation. So to summarize my results, we can see that squid skin does have a distinctive absorption spectrum, and that there are clearly distinguishable emission patterns caused by stimulation or excitation in the ultraviolet range, which are distinctive from the emission spectra that are presented by the water that they are contained in. Therefore, we can see that clearly there is something interesting going on with squid skin at these areas. Furthermore, we can tell by using confocal photoluminescence that indeed the chromatophores themselves seem to have certain particular properties, at least in excitation wavelengths of the green range, which cause them to fluoresce far more so than the skin that surrounds them. So what can we do next? Well, one of the things that I'm interested in trying would be further photoluminescence measurements using different excitation wavelengths in order to see if this is something that is consistent over a broad range, or if the idea of chromatophores simply fluorescing more than the skin is something that might be more contained. Additionally, I'd like to look at the impacts of other factors of light, such as polarization, intensity, and interference, as well as some physical properties which might change the optical properties of the skin, such as pressure. Additionally, I am interested in possibly looking at the pigments themselves, which are contained within the chromatophore, distinctive of the chromatophore as an organ as a whole, to see if perhaps the pigments might be able to show some different properties or confirm what we already suspect. And finally, I'm interested in looking at ways to better preserve the specimens that we're using so that we can keep them fresh and well preserved for longer. So what I've learned over this summer, I've learned a lot, but not just things about squid. I've learned how to use a variety of techniques and instruments, and I've also, yes, learned how to dissect a squid, which is something that you probably will never forget how to do. Or doing. But really what I think is most important is that I've learned some things about research as a whole, which have really changed the way that I think about it. I've learned that inventive critical thought is actually really important to the way that you look at and approach a project. And additionally, that when you occasionally find a stumble, a mistake, or a dead end in research, this doesn't mean the end of the world, but it simply gives you more information to approach the project with that in mind and perhaps try a different method. I've additionally learned that it takes a lot of work, really, to work on a research project. But ultimately, the rewarding feelings and the idea of the results that it gives you are definitely worth it. So I have a lot of acknowledgements, and I'd like to start by first really sincerely thanking my mentor, Viva Horowitz, as well as my PI, Professor David Oshlam, without either of whom this would not have been possible. I'd additionally like to thank EUREKA and Carrie, who's unfortunately not here, as well as the rest of the people behind EUREKA and the internship programs in general for making this an amazing summer and really helping me to get into research. I'd further like to thank Will, Lee, Joe, and the rest of the Oshlam group, who are all extremely helpful in some of the parts of this project, as well as DARPA, who is providing us with funding, Danny DeMartini, Claudia Gastin, and Livia Meze, who all helped me with some of the aspects of this project, as well as the Hanlon group at MBL and the WHO group at Harvard. So, any questions?
So you talk about preservation and wanting to extend it, but what is the lifetime of these samples, of those pieces of skin that you're working with? When do they stop being fresh, for example?
Okay, so the issue is that there are certain techniques that are employed often by biologists to preserve samples, but what we've seen so far is that the ones that we've looked at using to try and keep these structures preserved and from decomposing for a long time ultimately actually cause problems for us. For example, they induce fluorescence, which means that they're completely useless for optical testing. So basically, it takes about as long as you would expect seafood to be reasonably edible, which is not that long.
Yeah. I'm just curious, what do these chromophores are formed at the molecular level? Did you see anything at the molecular level? What are they formed of, the chromophores, the chromatophores? Sorry.
Well, I haven't done any looking at them specifically at a molecular level. I know that they are essentially like little sacs, which are tiny organs that have a bunch of pigments in them. And actually, one of the groups that we're working with is looking specifically at those pigments. But for the exact details of what's in them, I'm not really sure.
Is the process that they arise from like how they grow as the squid or other cephalopod grows, has that been looked at much? Like how they go from a collection of skin cells to a collection of skin and chromatophores in the skin?
Oh, you mean like embryonic development?
Yeah, has that process been studied, and do you think it would be very enlightening to study in the first place?
It might be interesting to study. I haven't heard about any studies being conducted on it. I do know that the chromatophores occur rather early because I've seen some videos of very tiny transparent baby squid like bumping into each other and turning on each other's chromatophores accidentally, which is actually adorable. But I don't think that, to my knowledge, there's no research looking at the embryonic development of chromatophores.
You were looking at, was it mostly octopus skin, or did
Squid
you?
skin.
Squid skin?
Yeah.
Have you tried, looking at other species skins within this cephalopod?
I've considered this, and I actually ended up going down to a fish market and picking up an octopus as well. The issue, however, is that ultimately the seafood that you can buy at a fish market is not really that fresh. What's happened is it's been caught. The fisherman's had it on his boat for a while. He's sent it into a company who essentially sends it out to all the stores around. So by the time you get it, actually, especially for squid, the chromatophores have actually ruptured. They've exploded, and the pigment is all over, which explains why usually if you see squid in shops, they're not really transparent. They're kind of reddish brown. That's from the chromatophores rupturing. So it kind of defeats the purpose until I'm able to find somebody who can provide me with really fresh samples of it to try and look into that, I think, right now.
Yes, so there's something I always wondered, and maybe you can answer. So when a squid kind of puts itself in a certain position and takes on the pattern, the colors and everything of the rock or whatever it is that it's laying on, like, so how does that work? Does the squid like look with an eye and then gets the signals to the skin, or does the skin kind of does it by itself? Like if you blindfold squid, could it still do that?
See, that's actually a great question because biologists don't even really know. There are some guesses about how it works. One of the really weird things is that cephalopods in general don't have very good color vision. In fact, they only really see in the green range, but they're somehow able to match colors that are outside it. So a couple of the ideas that have been proposed are, a lot of it's just pattern recognition. And additionally, it's possible, there's been some research that suggested that there are like proteins in some cephalopod skin called opsins that sort of respond to light of different wavelengths in different ways. So actually, we're not really sure.
Have you heard anything about how like the dynamic camouflage might be? Because I know they use muscles to control the saxi. Have you heard anything about how that might be translated to artificial use?
Engineer, well, I mean, you can think that it would be possible to engineer something that would act similarly to a muscle that could respond to, for example, a voltage in such a way that it would contract or expand. So that could be doable. And theoretically, you could also do that at maybe even a smaller scale to make a better camouflage.
This may be out of the scope, but has anyone ever, or have you heard of any studies where people have studied if a squid or octopus or something can mimic a non-natural object?
Well, so there's this octopus called the mimic octopus, which will actually imitate other animals in order to deter predators. And I think so far we know of a repertoire of about 30 or 40 animals that it can mimic. And it does it pretty convincingly if you look it up. It'll adapt the patterns as well as the rough body outline, or in some cases the actions of that animal. So that's about the best that I know of. I don't know of anything, like what do you mean by non-natural?
Kind of a way to probe his question is, could you study, like, does it just do certain types of patterns, or what if you threw it a non-natural pattern? Would it be able to adapt to that non-natural pattern and mimic that as well, right? So like, is it an adaptable transformation, I guess?
There's actually a paper about that where somebody did exactly that, and they put cuttlefish on like checkerboard backgrounds. And one of the things that was interesting is when you put it on a black and white background, it does adapt essentially what kind of looks like checkerboard coloration. If you shrink the squares down enough, then it doesn't perceive it as different light and dark patterns, but just a solid. But it does seem that there are, at least for cuttlefish, certain discernible differences in the way they'll respond to different patterns.
This is definitely an interesting study that you're doing. So, thank you for sharing.